retrovirus, you can then get it to go into hopefully the right cell at the right time. But the challenge is that you have to give the DNA, you want to get it into the target cells. So there's one issue of targeting. You also want to make sure that you don't overexpress this gene or you somehow give too much of that gene, because that could also cause problems in the opposite way. And it has to be delivered at the time that it's needed. So that's a lot of logistics to be handling. The current vectors, they use different types of vectors, but retroviruses are commonly used. The reason they're called retro is because they have RNA genomes and they get converted into DNA. So anybody have a guess? If we're going from RNA into DNA, what is the name of the enzyme that makes a copy of DNA from RNA? We've already talked about it was used to make cDNA. Yeah.
Reverse transcriptase?
Yeah, reverse transcriptase. So these viruses, they encode reverse transcriptase because the way that they replicate themselves is that they take their RNA genome, they make a DNA copy, they insert into the host. Their goal is to get into the host genome. The problem with this is that when retroviruses insert, it's completely random. It's a random integration. It goes anywhere. So it's not targeted, and that can cause problems. So I'll kind of talk about when they use this, there were problems that happened because of the random integration. So this shows what the idea would be for gene therapy. You would put your target gene, the human RNA here. You put it into the retrovirus. That virus infects cells, and then reverse transcriptase, which is encoded by the virus, will be used to make a DNA copy. The DNA goes into the nucleus and gets randomly inserted in. That's what you hope will happen for gene therapy. However, as you might imagine, not all diseases can be treated with gene therapy. So if you want to treat a disease with gene therapy, you first of all have to know what gene is mutated to be able to know what good copy to put in there. So this can only treat diseases where we have a lot of information about what genes are mutated. You have to know what is the healthy version of the gene so that you can put that in. And then finally, it typically is used to treat a loss of function mutation. So the protein function has to be modified to repair the defect, meaning that if I add extra copies, that's going to fix the defect. So usually the disease is caused by non-functioning proteins, and I'm just giving it good functioning proteins. It can't be used to treat dominant diseases, because in dominant diseases, that protein is going to exert its effect whether or not there's a quote-unquote good copy present. So there's limitation on what you can treat. So for a second, I want you to think, which of these diseases could potentially be treated with a gene therapy approach based on what you know. So discuss with the person next to you, which on the list is most likely a good candidate.
Sickle cell.
Sickle cell?
Maybe cancer.
Yeah, that's kind of what I thought. I mean, obviously you can't do that to also diabetes.
Yeah.
So any ideas?
That we
Which
know.
one?
Yeah, that's true. Yeah.
Sickle cell anemia, everybody agree that it's caused by a single gene. We know what the gene is, we know what the healthy copy is. Why couldn't you do cancer? I mean, what's the reason why that's not a great candidate?
I mean, because cancer, it's a mutation in a random part of the gene, so you don't know which part of the gene it's going to.
Yeah, a lot of times we don't know. And actually, it's not usually just one, right? There's usually many mutations. Diabetes is one where although we know some of the genes that are implicated, we don't know all. And autism is one where there's still a lot of unknowns about what genes are implicated, and it's not a single gene. So there's sort of the limits with this. With the initial trial that they did to treat skin, they basically got stem cells from the bone marrow of the patients. And actually the initial trial that they did, I have the numbers here, it was 10 boys under the age of 1 that were selected to be part of this study. They took stem cells out, they had that retrovirus with the correct, the normal functioning copy of the gene. They put that into the virus, infected the stem cells so that they're doing outside of the body, and then they implanted those cells back into the bone marrow so that that stem cell population would replenish and they would have the correct copy. Now with that, 9 of the 10 after this treatment had normal functioning T cells, so that seemed like a success. But then it turns out that, I have the numbers here, 4 of those 9 developed leukemias when they were still children. And the reason for that, so leukemia is a blood cancer. The reason for that is that that gene had integrated randomly in a place where it was being overexpressed. So it got expressed at too high of a level, therefore causing proliferation of the T cells. So many of those children actually ended up dying from this gene therapy treatment. And so obviously we have to be able to target, if you could specifically target the correct copy to the actual location of the gene, that would circumvent a lot of these problems, right? So that's the idea behind gene editing. So kind of the very last topic that we can talk about is genome editing. How many people have heard of CRISPR before? Probably everybody has. Before there was CRISPR, there were other ways that you could edit the genome, but none of them were as effective as CRISPR. So obviously, this was discovered in 2013. I remember, because I was still in my postdoc at that time, and the very first paper that came out, this was like extremely exciting for everybody in the scientific community because it meant that you could change DNA really easily. Well, not easily insofar as there's a lot of logistical details, but that you could actually modify DNA is revolutionary because you can actually fix it at the source instead of trying to, like RNAi, knock down a protein, or gene therapy, try to deliver a gene. This actually goes in and fixes the DNA so that you get rid of that mutation. So these are 2 of the scientists that won the Nobel Prize for this in 2020. Emmanuelle Charpentier, she's interesting because she actually doesn't like the limelight, and then she discovered this system in bacteria which put her into the limelight. So this bacterial species, which is the bacteria that she was working with, and she discovered that they have this system. It's like an immune system for bacteria. And that system, then Jennifer Doudna took that out of bacteria and said, what if we use that? We can actually take the parts of that immune system and we can make modifications to DNA wherever we want to. So she kind of had the applied side of this. And just to briefly talk about how this works, I kind of have just to show you the growth of CRISPR by year. You can see how many patents there are. This is the different diseases that have been studied. Some of the biggest ones, so we do have neurodegenerative diseases. So anyway, this is like percentage of papers that are coming out. But this is what the CRISPR system is. This is what Emmanuelle was working on. She basically discovered bacteria have in their genome this section. The section of DNA is called CRISPR. So that section stands for clusters of regularly interspaced short palindromic repeats. That was in the genome. So what was that section doing? It turns out that in bacteria, they have, whenever they get infected with a virus, they take a copy of the DNA and they insert it into this locus, into their own bacterial genome. What this is like a vaccine card. So whenever you get an infection, right, in our cells, in our immune system, we remember that infection later on. So if you get a vaccine, you're delivering this little signature so that your immune cells will recognize it later on. So bacteria do the same thing. They take the DNA, they deposit it. Then from this locus, they use transcription to make CRISPR RNA, and they process these little sections that all originated from different viruses. They put them into this protein that's called Cas9. And the Cas9 is like the effector T cell. So it's basically this for this, whatever that signature DNA is, if it ever finds that DNA again in the cell, it knows that this was a virus that once infected, so it's gonna get rid of it. So what it does to get rid of it is it chops it. If it ever sees that DNA again, a match, cuts it in half so that this virus can't infect the cell. So you can kind of see how this is similar to our immune system, right? We have B cells that are looking for, they're looking for a signature. If it ever sees it again, it's gonna mount a response, and it's actually gonna get rid of that pathogen. So this is all the pieces that exist naturally in bacteria. What Jennifer did with this is to take Cas9 out of bacteria and basically create the targeting RNA. All you have to do is create an RNA that is going to match the sequence that you want to cut. And if you put Cas9 with that CRISPR RNA, it'll cut it. Now we generate, when you cut DNA, a double-strand break. So what are the two ways we fix double-strand breaks that we talked about in DNA repair? Yeah, so one option, just let the cell take care of it. Most likely it's gonna fix it with NHEJ, which means you're gonna induce some mutations right at the junction. So sometimes when people use CRISPR, they just let the cell fix it, and usually that actually will put mutations in the gene. But let's say that our goal is actually, we have sickle cell anemia, we wanna fix it, we wanna get rid of that mutation. You can also supply the donor DNA. If the donor DNA is there and it matches the sequence of this double-strand break, the cell will use that donor DNA to fix it. So you can actually tell the cell how to fix it. So this seems like great, right? The only problem that we have with this is we do have some off-target effects where, you're supposed to cut this piece of DNA, but you also might recognize other pieces of DNA, so you can induce cuts elsewhere that you didn't intend. And the other thing is, it's hard to make the cell choose this route. So sometimes it still fixes it with NHEJ. So they're kind of working out the details of how to do this. But what can we do with CRISPR? There's a lot of different things that have been done already. So there's been work to target integrated HIV. So some individuals have long-term HIV where that virus is integrated. They've been able to successfully get rid of it by targeting the integrated locus. CRISPR is used in agriculture. They're working on fixing lots of different diseases. I showed you all the different diseases they're working on. But if the fear is that if we have this power that we can change DNA, what happens if we start modifying human embryos, right? And actually this has already been done. So how many of you have heard about the twins that were born in China that had modified DNA? So a doctor, what's his name, Dr. He? Anyway, I have to look it up, the details again. But he did actually go in and modify genes, and then twins that had been modified were implanted and they were born. And in the scientific community, he faced, I don't remember exactly if he's in jail or not, but anyway, repercussions for this because we don't understand the extent of this. And doing this in human embryos, the scientific community agrees that this is nowhere near ready to be and there's a lot of questions whether we should ever be modifying human embryos and changing the DNA. So, okay. Yeah, and I know there's been worldwide conventions about sort of ethics of doing this, and should we come to some kind of a agreement about what we can do and what we should not do. So when we get to genetics, we will talk about ethics of different biotechnology things, and so that's one of the topics that we're going to discuss. I think it's next week we have a genetics discussion. So anyway, that's all we have time for today. Anybody have questions about CRISPR? Okay, so you should be familiar with kind of the steps. Like these two slides are the two slides that you should kind of know how CRISPR works and how we can use CRISPR to modify DNA. But other than that, that's it for today. I actually have to run downstairs at 2 for a meeting, so we can end class and I'll see you all